So basically, we have discussed two basic models. One is lock and key hypothesis, and second is induced fit. In the first model, actually, we have studied in our last class. The everything is fixed. So just like a lock and key, key is the substrate, lock is the enzyme. So that the each enzyme is having a specific substrate and it fits very well into the active site. So active site is the site present over the enzyme. Okay. So suppose this is an enzyme. So it is having some site where this particular substrate can come and fit. So this is actually the best model, which is showing that this is a lock and key hypothesis model actually. But the second model, that is called induced fit model, according to that model, as we have discussed in our last class, the binding of substrate induces some conformational change. Okay, so everything is not fixed. Instead, because the most of the enzymes are made up of proteins, so they are flexible actually. They can change their shapes. And most probably this change in shape is a result of binding of substrate. So once the substrate is able to bind this particular enzyme, it is making some conformational change in the enzyme itself so that this enzyme can more efficiently interact with the substrate. It can make more interactions. And we know that most of the interactions between the substrate and enzyme are non-covalent kind of. So the more number of interactions means there is more binding energy, and enzyme is able to catalyze the reaction by lowering the activation energy of the reaction. And we know the activation energy is a kind of barrier which will inhibit the substrate as to conversion of a product. Okay, so it is required that when the substrate is going to convert into a product, so activation energy should be lowered. Okay, so this activation energy is called EA. It is represented by some EA value. Okay, so it should be less so that more and more substrate can cross the energy barrier. So this enzyme is able to bind to the substrate with more number of interactions, mean more binding energy will release, and this will, the energy will be used to lower the activation energy of the reaction, because energy is not coming from anywhere else. It is itself the binding energy between the substrate and enzyme, which is being used to lower this particular activation energy, or to provide this activation energy so that the enzyme is able to convert the substrate into the products. And the products are released. The second thing which is most important in enzyme catalysis is that when the product is released, there is no change in the enzyme. So enzyme will remain as such. There is no change in the enzyme. Okay. Generally, most of the enzymes, not most of the enzyme, almost all the enzymes increases the rate of reaction. We know this, but there is no change in the equilibrium of the reaction, means there is no change in the concentration of substrate, in the concentration of product. Okay, so equilibrium will remain as such, means jahan pe hai wahi rahegi, but there is a change in rate of the reaction. Rate of reaction is generally increased in enzyme catalyzed reactions. Any catalyst can increase the rate of reaction, but enzymes are specific. We discussed this also in today's class, how they are specific, because in evolution they are designed in such a way, because their active sites are designed in such a way that most of the amino acids which are involved in the catalysis are close enough so that they can interact more efficiently with the substrate, so that they are able to convert the substrate into the products. So this also we will discuss. Fine. So this is a brief introduction. Now, before starting today's class, I will also let you know that in today's class, we are also going to discuss about various mechanisms of catalysis. So what are the various mechanisms of catalysis? So let's directly look into the. So this book is White and White, fourth edition I'm having. So the catalytic mechanisms are actually only 3 catalytic mechanisms are shown in your Pathfinder, but in this book, when you read this book, so actually we will discuss all the 6 ways of these catalytic mechanisms. So these 3 actually also included in your Pathfinder. If you have Pathfinder, I hope most of you have Pathfinder Part 1. So the catalytic strategies, okay, so this is given. These strategies are commonly, what happens actually, the one enzyme can employ more than one strategies. It is possible also, but at least most of the enzyme, so the enzyme commonly employ one or more of these strategies, which are given in the white or in this particular book, to catalyze specific reactions. And you know, the enzymes are very specific catalysts. They bind to the substrate very efficiently and specifically. Okay, so I will tell you the two basic ways by which these enzymes are able to recognize the specific substrate. So, okay, so geometrically also, stereospecificity also recognized in these substrate bindings. Okay, so that the enzyme can make the maximum number of interactions with its specific substrate. So that's why it is able to differentiate between different substrates. Okay, so they can employ one or more strategies to catalyze specific reactions. It is not always necessary that each enzyme utilize only one. It is also possible they can employ one or more strategies out of these followings. Among the best characterized mechanisms, so there are three actually, which are the basic mechanisms in most of the, means in the three classes actually most of the enzymes will be covered. Actually, we will see some of the examples. These include acid-base catalysis, metal-ion catalysis, and covalent catalysis. Now, a combination of the several catalytic strategies is usually employed by an enzyme. This is very important. Actually, this also we had discussed in the first line. A good example of the use of both covalent catalysis and a journal acid-base catalysis occur in the case of chymotrypsin. So there is a family of enzymes or proteases which are called serine proteases actually. So these chymotrypsin is a family member of large family that is called serine proteases. We will discuss serine proteases also. Okay, so first of all, there are three actually: covalent catalysis, acid-base catalysis, and there is metal-ion catalysis. Okay, so first of all, actually we will go in a sequence actually which is given in your standard book. So white and white is saying, first of all, so before starting these catalytic mechanisms, some important information I want to share with you. Okay, so enzymes, as we have seen, we all know about enzymes. They causes a rate enhancement. They increases the rate of reactions, and this rate increasement or rate increment is actually the several order of magnitudes actually. So which is orders of magnitude greater than those of the best chemical catalysts. How it is possible? So a question should come into our mind that catalyst, general chemical catalyst, also having the same strategy, they also increases the rate of reactions. But what is so special in an enzyme or a biocatalyst that it is always better so than a chemical catalyst? So the answer is actually enzymes have a characteristic that are not shared by the substances of non-living origin. So if some chemicals catalyst, if you take some chemical catalyst, some metal, for example, and if you take a biological catalyst, which is an enzyme or a protein, they obviously differ, and you always find that the biocatalysts or the enzymes are better than chemical catalysts in several ways. So to this day, there are few enzymes for which we understand more than cursory details how they achieve their enormous rate acceleration. Actually, also today we have studied so many enzymes actually, but we know everything only about few enzymes actually. So based upon which, based upon this particular knowledge which we have, is only over few enzymes we have studied in detail actually. So so the the enzymes are simply better designed. So in evolution actually this thing has happened that nature has designed these enzymes in such a way that they are having active sites so beautifully designed that all the amino acids which are required for their reactions, they are very close proximity. So we will see in these basic mechanisms actually how these enzymes active sites are having amino acids in very close contact so that they can interact more efficiently. So one thing I want to show you before we start this, see this for example. So this is an enzyme. So I'm showing the RNase A actually. This is RNase A. I will also tell you the reason why I'm showing RNase A first, not anything else. For example, I can also show chymotrypsin or lysozyme or some other enzyme, but I'm showing RNase A. So there is a reason behind this. I will tell you later, but before you look that this is a protein, okay? And this protein is made up of amino acids, and you can see the side groups of these amino acids. Suppose there is an amino acid. So let me, oh, where is the format? Okay. So suppose this is an amino acid here. So what kind of amino acid it is? If it is having this imidazole ring, obviously this is histidine. So suppose this is histidine in the side chain, and histidine is represented by an H. Second histidine in the same enzyme, RNase A. So what is the function of RNase? To cleave the RNA actually. So second histidine or H is here actually at this particular position. Okay, so you can see this is a chain of amino acid. There are several different amino acids also in between. So this is made up of amino acids. This is a protein. So this RNase A is having so many amino acids, but you will see that this is 12th number histidine and this is 119 number histidine are very close proximity. They are in close proximity so that they are able to degrade the RNA substrate. So an RNA species coming from outside, okay, so it is having some kind of general acid-base mechanism, and therefore it is able to cleave this particular substrate. In this case, the substrate is RNA itself. So that's why these two amino acids are in close proximity. So what is why we are discussing this? I want to tell you that enzymes are so much better design. They are having a better design in comparison to the chemical catalyst, so that their functional groups which are involved in the catalysis mechanism or catalysis reactions, they are in close proximity. Okay, so evolution have made them so efficient in this way actually. Now, before telling everything about RNase A, now I will tell you the reason why we are discussing RNase A first. Okay, so first of all, sorry. So first of all, we are going to discuss acid-base catalysis. So this is an example, RNase A is an example of this particular mechanism. This is the first reason. And the second reason is given in this particular PDF. Sorry. So when we were discussing about enzymes, certain points we have skipped actually. So I will cover them also in today's class. So although enzymes are subject to the same laws of nature that govern the behavior of other substances, they differ from the ordinary chemical catalysts in several important aspects. One is higher reaction rate. So generally the biocatalysts are having reaction rate better when we compare them with chemical catalysts. Second thing, we know they are working under milder reaction conditions. So generally they are working better in cytosol because there are lots of protein in the cytoplasm, and the cytoplasm is having near neutral pH. Cytoplasm is exactly having 7.2 pH. So generally they require, and the temperature is also within the cell is around 37. Third is greater reaction specificity. If we compare the bicatalyst with the general chemical catalyst, they are having better specificity. This better specificity may be contributed to the stereospecificity or the geometric arrangement of substrate with the enzyme itself. Something is given, for example, reaction specificity. So, for example, enzymatic synthesis of a protein over the ribosome, or this polypeptide consists of over 1,000 amino acid residues are made, but they are error free. There is no, they are very accurate, or there is no mistake actually. So, thus you can assume how specific they are. Okay, so also there is a thing, capacity for control. So these enzymes are able to regulate their synthesis or degradation within the cell. Okay, so the mechanism of these control processes include, these are undergoing some allosteric control, some covalent modifications. You know, these covalent modifications, phosphorylation, dephosphorylation is the best way, but also some other enzyme undergoing glycosylation event, for example, addition of glucose residues or some other groups over them so that they can be covalently modified. Okay, so allosteric regulation, covalent modification of enzymes, and the variation in amount of enzyme synthesized. You also know that in certain pathways, their synthesis can also control in response to the rate limiting step, which is generally the first step in a biochemical pathway. So suppose here we are having an example. So in your Pathfinder only, they have given this particular model where they have shown there are enzyme 1, enzyme 2, and enzyme 3 catalyzing this particle biochemical pathway. So the first step is generally the rate limiting step. So the product, which is the D of this particular metabolic pathway, is actually when present in excess, it will go and block the first enzyme. So this is called negative feedback mechanism of inhibition. Also, there are feedback loops which are able to inhibit the reactions or the synthesis of an enzyme or degradation of some metabolite at various steps. Okay, so this is kind of regulation which is shown by the biocatalyst or enzymes. Then so this is all about the initiation of the enzymology. So the research actually, from where we are able to identify that the enzymes are actually working to degrade. So there are research basically over fermentation. Okay, so fermentation, generally alcohol fermentation, for example, is widely considered to have begun in the 1810. This particular guy, Joseph Lucas, determination and production of ethanol and CO2 when the sugar is decomposed by some yeast. The same observation in 1835 with Burgellus, first general theory came into existence, and this is called the chemical catalysis theory, which point out that an exact or the extract, actually extract of malt, which is known as diastase. So this called this diastase. Now we know this particular enzyme diastase is also known as alpha amylase, is able to degrade the sugars which are present in that particular extract. Catalyzes the hydrolysis of starch. So the starch is present in plants actually. Okay, so more efficiently than those of the sulfuric acid. Sulfuric acid is also able to degrade or hydrolyze the starch, but it is not so much efficient in comparison to alpha amylase. It is less efficient actually. So this biocatalyst was found to be more efficient when compared with sulfuric acid or some other catalyst, chemical catalyst. Yes, yet despite of these ability to mineral acid to mimic the effect of the diastase or alpha amylase, it was the inability to reproduce most of the biochemical reactions in the laboratory. So actually, scientists at that time in 1835 or around 1835, when Jacob Burgellus was working over this particular extract of the yeast cells, so most of the scientists at that time, specifically this one, Louis Pasteur, gave a theory that is called vitalism. According to him, he thought that most of the biochemical reactions cannot be mimicked in the laboratory. They cannot occur outside the animal cells. And in the mid 19th century, he proposed a process of fermentation could only occur in the living cells. So This is called vitalism theory, that the processes or these chemical reactions can only occur in the living cells. They cannot be done in the lab or in the test tubes. Thus, as was common in his era, Pasteur assumed that the living systems are having some vital forces or some extraordinary powers to convert these, to catalyze these reactions actually. Okay. Only then another scientist, we have discovered this actually in our classes, Edward Buchner, obtained a cell-free yeast extract that could carry the synthesis of ethanol from glucose. That's called alcoholic fermentation. So actually, when this 1878, he was able to give a term, and another scientist, Kuhne, who gave the term enzymes. Okay. Then this lock and key hypothesis came into the existence. Jacqueline urease have been discovered first. The first enzyme which was crystallized was actually this urease, which was extracted from the jack beans, from a plant species in 1926. But okay, so the first crystallized protein and the crystals of this particular protein was formed, but actually these are not pure proteins or crystals. Therefore, some other scientists, John Northrop and most units have make some other crystals or some other digestive enzymes, crystalline pepsin, trypsin, chymotrypsin, and so on. Okay. Now, then in 1963, actually 1930s, it was widely accepted that the most of the proteins were actually enzymes, or the enzymes are mostly proteins, whatever you say. Then in 1963, the first sequence of amino acid sequence of an enzyme came into existence that is called bovine pancreatic ribonuclease A. That's why we are discussing ribonuclease A or RNase A first, okay? Because it was the first protein to be sequenced, whose amino acid sequence, this was the first enzyme whose amino acid sequence described initially. And the second important enzyme is lysozyme. In 1965, we are able to decode the X-ray structure of this particular enzyme. So we will discuss both today. One is lysozyme, one is ribonuclease A. So, but both are different. They are having different strategies of their catalytic mechanism. So ribonuclease A is having some general acid-base mechanism of their catalysis. So first of all, we will discuss ribonuclease A. So we are coming back to this particular thing in which we are having ribonuclease A also. Sorry, it is not moving. What's wrong? Okay. It can go up, not moving down. Okay, fine. So first of all, we're moving to, okay, so before starting ribonuclease A, let's look into the mechanism. So general acid-base catalysis, what is this actually? So the acid-base catalysis involve a proton transfer. So some of the protons, H positive ions, get transferred from one substrate to other. Okay, so in fact, from some water molecules or over the water molecules also. There are two kind of acid-base catalysis. One is called specific and general acid-base catalysis. In specific acid-base catalysis, hydronium ions or hydroxide ion act directly as an acid or base. So you can also get this one from here. So this is acid-base catalysis actually. So there are 6 types. So covalent catalysis, which is either nucleophilic or electrophilic. Acid-base catalysis can be of 2 types, general or specific. Actually, general is actually most of the enzymes follow this general way of, for example, lysozyme itself is having this general way of acid-base catalysis. Okay, so some other proton donors. Okay, so there are 2 ways. One is specific, one is general. If it is specific, so the hydronium or hydroxide ions are itself the proton acceptors or donors. It is being determined by the pH. and there is no effect of the buffer concentration in this particular class of acid-base catalysis. This is specific class. And then general one, so some other different proton acceptors or donors are being used. These are generally the ionizable R groups or the cofactors within the protein or enzyme, mostly in enzyme catalyzed reactions. But buffer concentration is going to affect the rate of reaction in this general acid-base catalysis. Okay, so this is the basic difference. This all is actually written in your Pathfinder also. If you look into this, there are two kind of acid-base catalysis. One is specific, one is general. In specific acid-base catalysis, hydronium ions or hydroxide ions, which are H positive and OH negative respectively, act directly as an, so H positive is an acid, OH negative is a base. These groups are used in general acid-base catalysis. Donors and acceptor proton are other than the hydronium and hydroxide ions. So one is specific. In specific, the hydronium ion and hydroxide ions are being used as donors or acid or base groups, whereas in general, they are some different molecules. Mostly they are ionizable functional groups of amino acid side chains in the protein or enzyme itself or cofactors. So they are acting as like acids or bases. The functional groups such as imidazole ring of histidine, hydroxyl group of, so hydroxyl is present in 3 amino acids. One is serine, threonine, and tyrosine. Carboxyl group either in aspartate or glutamate. So hydroxyl groups either in cysteine, for example. Amino group is present in, okay, so this is amino group, either lysine, which is having amino group, or the amino group can also be present from amide group, CONH2, which is present in asparagine and glutamine actually. Okay. We know lysine is having epsilon amino group. Phenolic group, which is present in some aromatic amino acid, for example, phenylalanine or okay, phenolic group is generally present in phenylalanine, tyrosine, but phenylalanine is the best example. So these are acting like either acids or bases, whichever we have discussed. So if these amino acids are present in most of the cases, for example, in ribonuclease A, RNase A, imidazole is being used as uh, either an acid or a base. Actually, the two histidine molecules, either a 12th position or in 119 number position in the enzyme itself, we will discuss as well. As far as the enzyme catalyzed reactions are concerned, only general acid-base catalysis can occur. A common example, okay, so in enzyme catalyzed reactions, mostly this is general class of acid-base catalysis. So we are having two enzymes, one is lysozyme and one is RNase A. Okay, so RNase A first because it was first enzyme whose complete amino acid sequence was decoded. Okay, so common example of general acid-base catalysis, like hydrolysis of peptidoglycans, which is done with the help of lysozyme. Specific acid-base catalysis is defined by the pH only. So the first one in which the hydroxyl hydronium ions or hydroxide ions are being used. And this there is determined by the pH, but there is no effect. But the buffer concentration has no effect. So you should underline this line actually. So there is no effect of this concentration. Okay, so buffer concentration is not affecting, but the buffer is having specific pH. Therefore, this is determined by the pH of that buffer, not by the concentration. While the general acid-base catalysis, however, buffer is going to affect the concentration because the buffer concentration is important because the molecules which are present in the buffer, their concentration is going to donate or accept a proton, and therefore they are helping in transition state and thus affect the reaction rate reaction or overall reaction. Now we will go to this portion where they have discussed about these enzymes. So ribonuclease A is given over this page number. This is RNase A. Okay. This is also discussed in your book, White and White. Okay, so important points are also given. Okay, so you can also look at over these highlighted text actually. So if you want to define the catalysis in 1.5 line, you can say a process that increases the rate of reaction without affecting the equilibrium. Okay, so generally it increases the rate of reaction by lowering the activation energy. This is called EA, which is a kind of barrier. So it should decrease. Okay. It's just like if you decrease the bar, okay, so in a high jump, so more number of athletes will cross the bar actually. So similarly, if there is less kinetic energy, less activation energy of the reaction, so this barrier is crossed by most of the substrates. So a large number of substrate will be converted into the product. Similarly, so it require make an enzyme so powerful catalyst is actually the result of two related properties. One is specificity of the substrate binding combined with, so they are very specific for in binding to their substrates combined with their optimal arrangement of the catalytic groups. Second is the design of their design, or you say the optimal arrangement of their catalytic groups or the amino acid side chains so that they are able to catalyze the reactions more efficiently. So the first thing actually it is discussing about the specificity of the, so from where the specificity is coming from the stereospecificity. Enzymes are highly specific both in binding the chiral substrates or in catalyzing their reactions. The stereospecificity arises because enzyme, by virtue of their inherent chirality, the protein, because the protein is also made up of L amino acids only in the case of humans, for example, or in eukaryotes. So forming asymmetrical active sites so that these asymmetric active sites can only bind to the specific substrates. Okay. Now, so this we have discussed. Also, this is a product of eons of evolution, large number of years actually, which this evolution arranged these functional groups in such a way that they are very efficiently fine-tuned, so they are very efficiently performance of the most of the enzymes. Okay. Now, so we were discussing acid-base catalysis, in which it is actually the process in which partial proton transfer from a Bronsted acid or base. This term actually we have studied in our 11th or 12th Bronsted acid-base concept. So here also we are going to discuss the same. So this Bronsted acid is a species which is going to donate the protons. And Bronsted base is a kind of species which is going to accept the protons. So everything is revolving around these protons as positive ions. And the second thing is because it is an enzyme, lowers the free energy of the reaction transition state so that it can be converted into the product more efficiently. So this is a basic reaction. This is an uncatalyzed reaction. Okay, so first of all, this is showing an uncatalyzed reaction. You can get it from here. This is uncatalyzed in the absence of any enzyme. Then there is general acid catalyst and there is a general base catalyst. These two catalyzed reactions, when compared to this one, they will happen, but because the uncatalyzed reaction will take more time when compared to the catalyzed reactions. Okay, because these uncatalyzed reactions are having some high energy carb-anion like transition state. So if you look into this diagram, this A is an uncatalyzed reaction in which it will lead to the formation of a transition state, and this transition state is having high energy which cannot be crossed directly. Therefore it will take more time and the reaction rate is very slow, you can in other terms. Okay, whereas a proton donation to the oxygen atom, however, reduces the carbanion character of the transition state, therefore catalyzing the reaction. If some proton donation can happen Okay, either a acid or a base can do this job where it will donate the proton to the oxygen atom so that the rate of reaction will increase because there is a less probability of formation of this transition state. So ultimately, the rate of reaction will be high. Okay. Then there is a best example where you can see this general acid-base catalysis. If you have studied about the mutarotation, which are the alpha linear form and beta conversion of glucose molecule itself, so this actually has been recorded with some uh, instrument which is called polarimeter, which will measure this specific rotation. So if it is the solution is having specific rotation around 112.2 degree, it is showing the most of the species in the solution are alpha glucose. If it is having a rotation around 18.7 degree, it is showing it is beta glucose, and somewhere in between there is a linear form. So this mutarotation is a acid-base catalyzed reaction in our body. Then there is a second enzyme we were discussing actually. So this is called RNase A. So many types of biochemically significant reactions are susceptible to acids or as well as bases and their catalysis. These include hydrolysis of peptide bonds, esters, the reactions of phosphate groups, tautomerization reactions, and the addition of carbonyl groups. All these reactions are following this trend of acid-base catalysis. The side chains of the amino acid residues, for example, aspartate, glutamate, histidine, cysteine, tyrosine, lysine, they are having pK value very near about to the physiological pH range. Therefore, they are very actively participating in these acid-base catalysis reactions. Okay. So, indeed, the ability of enzyme to arrange the several catalytic groups about their substrates make the considered acid-base catalysis a common enzymatic mechanism. So, most of the enzymes which are involved in these reactions are actually following acid-base catalysis. Okay. Here they have written about RNase A. So this has been isolated from bovine pancreas, which is a kind of degradative enzyme, RNA degradative enzyme, which will cleave the RNA species, provide an eliminating example of the enzymatically mediated general acid-base catalysis. So this RNase A, which was also the first enzyme to be whose amino acid sequence was sequenced. So here, it hydrolyzes RNA into the component nucleotides. How it is able to do this? So this is a RNA molecule which is having a phosphodiester bond. So this 3' OH group and 5' phosphate is making this phosphodiester bond. And this 2', we know this is RNA, therefore the second prime of this ribose sugar is OH actually. So this is the basic difference between the RNA and DNA. RNA is having ribose sugar, DNA is having deoxyribose sugar. So if it's deoxy, so this oxygen should be deleted from here in DNA. But this is RNA, so it is having ribose, simply it is having OH here. So what will happen actually, how this is going to catalyze this reaction, this OH group will nucleophilically attack this phosphate group because it is having a positive charge surrounded by negatively charged oxygen or electronegative oxygen atoms. Therefore, it will develop some positive charge so that this negative OH group will attack this positively charged phosphate. Therefore, it will make something like this. It is a 2', 3' cyclic nucleotide intermediate. So it will result in the breakage of this particular phosphodiester bond. This will be broken from here. So this break will generate some OH group over this 5' terminus of this residue. So the 3' end of this molecule will have a 3OH hydroxyl group, whereas this will form a cyclic intermediate rest chain. Now what will happen, a water molecule will come and it will release some H positive ion or proton. Okay, so this will then cleave this particular intermediate, cyclic intermediate, by providing the H to this. So this will be OH, and this will generate this particular. Now this 3' ribose sugar, carbon of ribose sugar, will have a phosphate group, and this 5' of the broken chain of RNA will have a hydroxyl group as a result, and there is no change in the enzyme itself. Okay, so how this is happening actually, now we will look into the mechanism of this particular enzyme in detail. So it will show the RNase A will show a maximum rate of reaction around pH 6. Each enzyme is having a specific pH over which it is having maximum activity. So this is having little acidic value. So this is having peak around 6. Analysis of this curve together with the chemical derivation and the X-ray studies. So when we did X-ray study, it indicated that the RNase A, which is having two essential histidine residue, one is 12th and the second is 119 number of histidine. So all these two histidine actually having imidazole ring as a side chain of amino acid, they were involved in the reaction. So first of all, this histidine, which is small in number, means 12th, or I can say N-terminal histidine, is 12th number histidine is acting as general base because it is present over the N-terminal side of the enzyme amino acid sequence. So it will act as general base. It will remove the proton from this particular hydroxyl group, okay, OH group. So I have said the proton from the RNase 2' OH position, and thereby promoting its nucleophilic of addition of the other molecule. Okay, so see here. So this is the 12th number histidine, which is having imidazole ring. Therefore, it is having a negative charge here. It will donate this to this particular molecule. Therefore, it will make a hydroxyl radical here. Okay, so this will be OH negative. So first of all, we will look into the pathfinder, what they're saying. So they are saying the same thing. Actually, there are various species of the RNAs, for example, RNase N, RNase 2, 3, RNase H, which will work on hybrid DNA RNA hybrids, RNase A, which we are going to study in this particular class. So all are different type of ribonucleases or RNases. They are the enzymes which catalyze the hydrolysis of phosphodiester bond in RNA molecule. So bovine pancreatic RNase A, we are discussing the most studied RNAs. We have studied a lot about this, so we have a clear knowledge of their mechanism of action. Catalyzes the hydrolysis of phosphodiester bond, then it is in the single stranded RNA itself. Between the phosphate group of pyrimidine nucleotide. You know, pyrimidines are CUT. Okay, there are 3 pyrimidines and an adjacent nucleotide to give a 3' phosphate and RNA fragment with 5' hydroxyl terminus. If it is not clear to you, it is giving 3' phosphate. So look into this. It will give a 3' phosphate. So this is the 3' position of this particular RNA, and 5' will be released with some hydroxyl group over it. So this is a result of this particular reaction. Now how it is happening? So we look into the mechanism now. So the imidazole group of histidine 12 acting as general base catalyst, we have discussed this. It will abstract the proton from this particular, so it will remove this H from this OH hydroxyl group. So it will abstract the proton, H positive will be removed, and the electron will be transferred to the oxygen itself. So it will generate some nucleophilic alk oxide anion, which can be a direct question. So you should underline this whenever this acid-base catalysis is happening in case of RNase A. So it will lead to the result, lead to the synthesis of a species over this 2' carbon of ribose sugar. This is called, is having nucleophilic properties because it will going to attack some nucleophile positive charge loving species because it is having some negative charge. This is called alk oxide anion. Okay, because this proton is being abstracted by this 12th number histidine in this, which is acting like a journal base catalyst. Fine. This alkoxide anion attack the neighboring phosphate over the 3' carbon of this same ribose sugar. So it will attack the positively charged phosphate because it will have a positive charge because it is surrounded by all the negatively charged or the electronegative oxygen atoms. So they will attract, they will remove the electrons from this bond. They will attract the electrons so that it will develop a positive charge, though delta positive charge. It is a small charge. Okay. So after attack, the cleavage of this bond, this is a phosphodiester bond, and the substrate 5' 3' phosphodiester bond yield a cyclic intermediate. Okay, so this cyclic intermediate will be formed when this bond, so it will attack this one. So this bond will be broken down, and this will these electron will be shifted to the acid actually. So it will act. Okay, so first of all, we are discussing the 12th number of histidine. So histidine 12 is acting as a base, general base, whereas this will act like an acid in the first step. Okay, so the released 5' alkoxide anion pick up the proton from the histidine, which is on the C terminus of the same protein or the RNase A. So this will act like journal acid catalyst. So in the first step, this is an acid, journal acid catalyst. This is a journal base catalyst, and they are going to degrade the RNA. Now in the second step, this will be reversed. Then it will become a base. This will become a acid. Now see, the 2', 3' intermediate hydrolyzes in the next step. So this hydrolysis reaction is being done with the help of some water molecule. So in this particular step, actually this is not shown in this particular PDF. It is very clearly shown in white and white that one water molecule is being added, and this hydrolysis reaction has result into the release of the product molecule. So here you can see this green color water molecule. So it will accept the this electron cloud from this particular histidine, which is 119 position, and this is 12th number of histidine. So it will going to accept the proton this time. So this will act like an acid, this will donate, so that will act like a base, and water molecule is helping this in the hydrolysis, so that the OH which is coming from the water will be coming over this 3' position of the ribose sugar, whereas the hydrogen which is coming from here, actually this is hydrogen which is donated by histidine itself, so it will come here. So this is, and the enzyme will be regenerated. There is no change in the enzyme, so it is not having anything. So, and the second thing you need to remember, this particular H. So in the first step, it is in red color, which is being donated to the this 5' position of this ribose. Okay, then it was transferred over the 2' position of this particular ribose. The new hydrogen which is coming and regenerating this imidazole is coming from the water molecule actually. Okay, so now, so the role of histidine 119 and 12 are reversed in this particular hydrolysis reaction or this particular step. So it is made up of two steps. In one step there it is a base, it is an acid. In the second step it is a base, and it will act like acid. So thus histidine 119, whose imidazole group is now unprotonated, abstract a proton from the water. So it will take a proton from the water and will act like a base in this way. Whereas the hydroxide ion, which is created in this step, attack the phosphorus atom in the intermediate. 2' oxygen is leaving the leaving group in this particular step retrieves the proton from the imidazole ion of the histidine number 12. So it will act like an acid. Okay, so that's how they will be having reverse functions in this particular step, and this will be generated. So is this clear to you? Are you able to understand how this RNase A is able to degrade the RNA species with its two active amino acid. One is histidine 12, one is histidine 119. So these are working as general acid or base catalysts. In the first step, the 12th number histidine is a general base. In the second step, it is a general acid. And same is the case with the histidine. In the first step, it is a general acid. In the second step, it is a general base. So they will act like this, and a water molecule is helping in this particular step, in this particular process. So before moving to other enzymes, for example, we are going to discuss others also. Is this clear to you all? I do not know how many are remaining. Only 3 students in class. Charan, Neetu, Trishla. So Trishla, Neetu, and Charan, is this clear to you? If not, I will. Okay. Otherwise, I will. Okay, I will tell it again. Actually, this is important. So you need to remember the very important things out of this text actually. So one is this particular steps actually. In the first step, this histidine over this N terminus, which is the 12th number histidine, is acting like a general base, whereas in the second step is general acid. So this you should remember. Second thing, it is abstracting the proton from this second prime hydroxyl group of this particular ribose sugar in the RNA itself, and it will generate some alkoxide anion. So this can be a direct question. So this alkoxide anion is being generated, which is having some nucleophilic function in this particular reaction mechanism. Okay, so finally it is able to degrade the RNA. Therefore, it is called RNase A. Second is, okay, so everything is given here. So the enzyme is coming into its original state after accepting one proton from the water. Okay, so this will be regenerated. There is no change in the enzyme because in any enzyme catalyzed reaction, there are no changes in the enzyme actually. Second type of reactions which are catalyzed by the enzymes are covalent catalysis. So in this particular catalysis, a CIF base which is having some double bond. So this is called imine bond. So if you know amide bond, which is a CONH bond actually, so this is having a positive charge here because valency of nitrogen is 3, but here it is bound with 4 bonds. Therefore, it is having positive charge. And carbon is actually bound to 4 bonds already. So this type of structure is called CIF base, which is present, which is formed when there is a covalent catalysis. So this transient formation of catalyst substrate covalent bond. This is a temporary covalent bond, but it will be formed whenever there is a covalent catalysis. Okay. Then this CIF base is being degraded. The formation and decomposition of this base occur quite rapidly, so that these steps are not rate determining step in the reaction sequence overall. Now we will look how actually this covalent catalysis is being happening. What are the basic steps? So there is a nucleophilic reaction between a catalyst and substrate, which is forming a covalent bond between the two. And there is a withdrawal of electrons from the reaction center. Now here the reaction center is made up of generally 3 amino acids. What are these 3 amino acids? HSD. So histidine, serine, and these aspartate. So you will remember it like this, HSD, which will make the charge relay system of that particular enzyme. So these 3 will be hydrogen bonded to each other so that they can form a charge relay system, CRS. So this will be formed out of this catalytic triad. Finally, there is elimination of the catalyst by these bicatalysts or enzyme will be removed or eliminated, and the reaction is Generally, the first step means the bond is being broken between the catalyst and the substrate. Okay. Now we are going into the detail of this particular mechanism. So something is given here, for example. Okay, actually they skipped this diagram from here, but we are having this. So first of all, I will tell you what is the covalent catalysis. So we are going a few pages back. So the process of covalent catalysis involves the formation of, if you're having the Pathfinder 7th edition, so you are having all this. Actually, this is given over page number 111, process of covalent catalysis involves the formation of reversible covalent bonds. So why is it called reversible? Because it is transiently or temporarily formed. Once this is formed, the reaction will over and then it will be reverted, means broken down. And this is found between the enzyme and substrate. Although the covalent interactions are temporary and the substrate is bound to the enzyme during the course of catalysis only, then it will be, this covalent bond is broken. After the completion of the reaction, the enzyme return to its original unmodified state, because we know in any enzyme catalyzed reaction there is no change in the enzyme itself. The proteolytic enzyme chymotrypsin is providing the best example of this particular class. Not only chymotrypsin, actually several other enzymes which are coming into the same class. For example, trypsin, elastase, all these are having the same catalytic triad, HSD. So they are making a charge relay system. So many will come in this class. Trypsin, elastase, thrombin, actually. Thrombin is involved in the clot formation or degradation. Thrombin, subtilisin acetylcholinesterase, which is also involved in the nerve transmission. Okay, so these are all coming in the covalent catalysis mechanism examples. So these groups, what type of groups? Nucleophilic centers are made up of amines, carboxyl, hydroxyl, imidazole, and thiol groups. These perform the covalent nucleophilic catalysis. Okay, and readily attack the electrophilic centers of substrate, forming a covalent intermediate. So obviously these covalent bonds are formed. And there is second type is, this is nucleophilic. The second type is electrophilic centers are formed. So the substrate include the phosphoryl groups. Now the groups or the amino acid side chains or R groups will change. So the enzymes which are having phosphoryl groups, acyl groups, glycosyl groups, they will act like electrophilic centers in this covalent catalysis. Whether it is covalent electrophilic catalysis or whether it is like a nucleophilic catalysis, as they're observed and usually involves enzyme adduct that generate the electrophilic center. Electrophilic catalysis generate electrophilic centers. Nucleophilic catalysis generate nucleophilic centers. So this you need to remember. And then we are having one example. Best example is chymotrypsin. Okay, so this is shown in your Pathfinder at page number. Okay, so we have page number 130. Okay, so chymotrypsin. Chymotrypsin, as synthesized in the pancreas, is a precursor called chymotrypsinogen. You know, some zymogens are present in our body, which are inactive forms of the same enzyme. So we will discuss one more figure in this particular context. Okay, so this is also given here. Sorry. Okay. Actually, we are having this also. You know, isoenzymes, which are the same enzymes, forms of same enzymes, different amino acid sequence, but they are catalyzing the same reactions. Then we are having zymogens. Zymogens are inactive precursors of the enzymes. So let's see first how this chymotrypsin is being formed. So it is formed from the one substrate, which is a zymogen, that is called chymotrypsinogen. And this chymotrypsinogen is being converted into the active enzyme chymotrypsin with the help of some enzyme trypsin. And trypsin is present itself as an inactive form or zymogen form, that is trypsinogen. Trypsinogen is being converted into the active form trypsin with the help of some enzyme entropeptidase, which is very important. So two enzymes which are going to, in the duodenum of your stomach, the pancreatic enzymes or zymogens, one is called trypsinogen, this one, chymotrypsinogen, prolastase and procarboxypeptidase are being converted into the active form by two enzymes, entropeptidase and trypsin. So entropeptidase, first of all, convert this trypsinogen into trypsin. Then this trypsin is going to convert all the inactive forms into the active form. For example, chymotrypsin will be converted into chymotrypsinogen will be converted into chymotrypsin. Prolastase will be converted into the elastase. Procarboxypeptidase will be converted into carboxypeptidase with the help of the trypsin only. Okay, so this trypsin and entropeptidase is going to convert all the zymogens into the active form, but mostly this trypsin is going to work in conversion of these three. Now, actually, this is given that this chymotrypsinogen is made up of 245 amino acid residues. So now you will need to look into the mechanism also how these zymogens are being converted into the active form. So, for example, many proteolytic enzymes, let's say chymotrypsin, trypsin, are initially synthesized in an active precursor, so that only when the need is required, they can be converted into the active form. Okay, so all these are proteolytic enzymes, protein degradation enzymes. Okay, so the specific cleavage is required at a specific site. If the enzyme is being cleaved, it will be converted into the active form. How this cleavage is going to help the enzyme to conversion into the active form? It will lead to some conformational changes in the active precursor that exposes the enzyme active site. So what is the difference between a zymogen and active enzyme? So this is a zymogen, trypsinogen, for example, and trypsin is an active enzyme. When this is being broken down at specific amino acid residues, so it will lead to the exposure of some of the active site residues, because the active site is made up of some specific amino acid R groups or the functional groups. So they will be exposed, so that after exposure, they will react with the substrate molecules, convert them into the products. Similarly, the one example is given here. For example, this chymotrypsinogen is made up of 245 amino acid residues, and they are connected with the disulfide bonds actually. So these are cysteine actually. So where they are actually these sulfurs are making disulfide bonds. So if someone asks you how many peptide bonds are there, so there are these disulfide bonds, not peptide bonds, and if someone asks you how many cysteine residues in the sequence, so 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Okay, obviously there are 10 cysteine residues. They're making 5 disulfide bonds. Now, in the chymotrypsinogen in the inactive state, there is no use of making these disulfide bonds, though they are helping in the folding of these particular proteins. But the trypsin will come and it will cleave this 15-16 number. Okay, so after cleavage of this particular peptide bond at 15-16 number, so it will be converted into an active form. So it will be called pi chymotrypsin. So question have asked from this particular diagram. Okay, so you need to remember whichever the first intermediate is formed, that is called pi chymotrypsin, is also having some activity, but it is not so much active as the alpha chymotrypsin, which is being formed in the second step of cleavage. So trypsin further cleave and it will cleave out 2 residues out of this. So 15-16 number peptide bond is broken in the first step, forming the pi chymotrypsin, which is active. Also in the second step, this will be removed, the 2 amino acids will be removed, which is 14-15 number amino acids. So this 14-15 number amino acids are actually S and R. So you can remember this by a trick. So this is called serine, this is called arginine, R. Also in the second step, not in the first step, there is no cleavage, further cleavage. So it is having less activity, though it is active. Then in the second step, when the pi chymotrypsin is being formed, this is also very active. The second cleavage is between 146 and 149. Actually, the two amino acids are removed, 147 and 148. So these two amino acids are being removed, and these two amino acids are actually, these removed amino acids are threonine T and asparagine N. So you can remember this particular sequence as star 10. So you can add some more letters to remember this. Okay, so here we can add STAR, and because this T we are adding, so this is T, and we can add E here. So this is called star 10. So the 10 stars are being removed from this particular sequence so that this can be activated. Also, sometimes the question coming like this, name these amino acids. So these amino acids which are being left actually over this active, so there are three chains now, A, B, and C. This may be a question, direct question. So the active alpha trypsin is made up of three chains, and these three chains are being connected by these disulfide bonds. Okay. So in this case, there are two disulfide bonds between B and C. There is only one disulfide bond between A and B. And this is actually, these two disulfide bonds are present within the chains actually. So you should also need to remember this also. And also the remaining amino acid, the 13 number position is called LIA, L-I-Y-A. Tyrosine is Y and alanine is A. So this is LIYA, which has been left, and STAR, and this has been removed. So hope this is clear to you all, so that we can move to the mechanism of this alpha chymotrypsin directly. So, okay. So we are discussing the chymotrypsin mechanism of action. So we are moving directly to the chymotrypsin part. So this chymotrypsin is being synthesized in the pancreas. Now it is clear to you how this in the pancreas, the precursor is called chymotrypsinogen, which is being degraded by the trypsin to make pi chymotrypsin and then alpha chymotrypsin finally. Alpha chymotrypsin is being formed from the pi chymotrypsin, and the two amino acid at the position 14, 15th number position has been removed out of the pi chymotrypsin. So these are S and R, whereas the other is from the 146, 147 number position. Finally, this is enzymatically inactive, and the alpha chymotrypsin which is formed is active. Now this active chymotrypsin is generated as a result of the cleavage of trypsin, is having four amino acids LIYA at cleaved positions and are connected by, and it is having three chains A, B, and C, which are connected by some disulfide bonds generated as a result of cleavage in three polypeptide molecule. This is A, B, and C interconnected by disulfide bonds. This enzyme catalyzes the hydrolysis of peptide bonds of proteins in a small intestine, or you can say in the duodenum part actually. It is selective for a peptide bond with an aromatic or large hydrophobic side chain that is either aromatic or a large hydrophobic side chain, whether it is tyrosine, tryptophan, phenylalanine, or methionine. This you have studied in your protein sequencing part, sir has covered actually. So this should be on carboxyl side of this particular bond. Then chymotrypsin also catalyzes the hydrolysis of an ester bond. How it is able to hydrolyze this ester, this peptide bond? Let's see the mechanism of action. So this is a serine protease. So serine proteases are a class of proteolytic enzymes whose catalytic mechanism is based upon the active site serine residues. Now look into this is an enzyme, chymotrypsin, for example, and it is having, there is an amino acid which is called serine 195 in this particular. So this serine may vary in position in different proteolytic enzymes, but in chymotrypsin, this is forming a catalytic triad or a charge relay system. It is made up of aspartate, D, histidine, H, and most important of all is serine, that is 195 number position. So This can be remembered as HSD, okay, that I told you. So this HSD is working as a charge relay system. So they are also connected via some hydrogen bonds. So these are hydrogen bonds, one hydrogen bond, other hydrogen bonds. Now, so this is very important. Serine 195, which is here. Okay. So the proteolytic enzymes having the same active site serine residue, this family also include the trypsin, elastase, thrombin, subtilisin, and acetylcholinesterase. This is being obtained firstly from the Bacillus subtilis, therefore called subtilicin. Also, it is being obtained from some other species of bacteria later on, but actually this is a kind of proteolytic enzyme which will degrade some specific protein. Okay, acetylcholinesterase is going to degrade the acetylcholine, which is actually a neurotransmitter after the neurotransmission. And other related enzymes may also come in this particular class. Chymotrypsin cleave the peptide bond. How it is able to cleave the peptide bond? Let's see. By attacking the unreactive carbonyl group with the powerful nucleophile, which is its serine itself. So this is a powerful nucleophile. The serine 195 is going to act like a powerful nucleophile. What is nucleophile? Which is a positive charge loving species. It's going to attack a positive charge because it is having OH. Okay, OH is O is electronegative, so it can attack some positively charged species. Residue located in the active site of the enzyme. So because it is present in the active site, so that is going to attack nucleophilic attack also. Nitrogen or the histidine or some other molecule in the protein. So let's see. Briefly becomes covalently bonded to the substrate, not only because it is a powerful nucleophile, it is going to make a covalent bond with the substrate. Therefore, it is coming under the covalent catalysis, forming an enzyme substrate intermediate. Treatment with an inhibitor that is called DIPF, diisopropyl phosphofluoride, is going to inactivate this irreversibly. So this can also ask a direct question from this. So what kind of inhibitor this is? This is irreversible inhibition. Because it will interact with this serine 195, it will make a covalent bond and this will not be broken, because we know the covalent catalysis involve the temporary formation of a covalent bond. It should be reverted back. But if the DIPF will react with this particular enzyme, it will permanently bond to this particular serine, so that it will allow the serine to react. It will not allow the serine to react at all in the active site of the enzyme. This chemical modification, this is kind of chemical modification reaction, suggests that this serine is very important to this particular enzyme in the reaction. Okay. So it is having some central role in catalytic mechanism of the chymotrypsin. Serine is a part of catalytic triad that we know. It also include the histidine and aspartic acid, aspartate. Highly reactive serial residue, that is serine 195, participate in the charge relay network with these two amino acids. One is histidine at 57th number position and aspartate at 102. Side chains of serine, that is OH, or in fact CH2OH, is a hydrogen bonded to this. Okay, so histidine 57, NH group of imidazole ring in turn hydrogen bonded to the carboxylate group of this aspartate. So they are also hydrogen bonded to each other, and therefore catalytic triad acts as a charge relay system. So this you should remember. Okay, so this is example of a whole group or a whole family. It's called serine proteases, in which there are so many enzymes: trypsin, chymotrypsin, elastase, thrombin, subtilicin, acetylcholinesterase, and so on. Are you able to get the chymotrypsin mechanism of action, which is an example of covalent catalysis? So that we can discuss others also. Okay, fine. So the third one is actually, okay, so before moving into the third one, so we also have one more example that is called lysozyme. So lysozyme, one question has been asked in GATE exam from the lysozyme actually. So I will also tell that particular question. So in that question actually they have asked this type of intermediate is formed in this particular reaction of lysozyme. So we know lysozyme is enzyme which will specifically degrade the beta glycosidic bond in the cell wall of bacteria. So it will make intermediate that is called carbonium intermediate. Okay, so this carbonium ion intermediate has been asked in one of the examinations. So you should remember this carbonium ion is formed in this particular reaction, okay, of lysozyme. So lysozyme is also called, as we have discussed about enzyme commission and their numbering, you should remember at least few examples. For example, lysozyme is having this number EC32117. This is easy to remember, 3 to 1. This is in decreasing order, and then 17. Also, many times they have asked 1.1.1, sorry, 1, and there is another one. So they have asked this about this particular enzyme. If you do not remember about this 1.1.1.1, so this is an alcohol dehydrogenase actually. So this alcohol dehydrogenase ADH, so you should remember at least few. So this is alcohol dehydrogenase. This is lysozyme is actually 32117, and we have also discussed about hexokinase. So okay, so actually this was the first enzyme discovered because the scientists were discovering the catalytic mechanism of the metabolic enzymes or some these are called fermenting enzymes, alcohol dehydrogenase or alcohol fermentation. So therefore, this was discovered first and named 1.1.1.1. So first class, it is involved in the oxidoreductases. This is the subclass, this is subclass, and this is the number which is given to alcohol dehydrogenase, which is actually the yeast enzyme, is obtained from the yeast actually. So you should remember these things before starting lysozyme. So we are now discussing the lysozyme. Lysozyme is having this particular number in enzyme commission. It is present in secretions, for example, in our tears, in our sweat. Also, it is found in the egg white. Okay, so this lysozyme can be obtained from so many places. Okay, now the first scientist, Alexander Fleming, discovered the penicillin. He also described this particular enzyme lysozyme in 1922. But the structure was described by another scientist, David Keighton Phillips, in 1965. It is present in egg white, so it is a monomer. It is obtained as 129 amino acid residues long amino acid protein. It cleaves specifically the beta-1,4 glycosidic bond has been asked in so many exams. So you need to remember this is very specific for beta-1,4 glycosidic linkage, which are present in the cell wall of bacteria. And it is present in between the NAM and NAG. So you should remember these beta-1,4 glycosidic bonds are present between the NAM and with the fourth carbon atom of the NAG itself. So if you look into the molecular structure of the cell wall of bacteria, they were made up of NAM and NAG alternative subunits of these two glycoside and okay, so they're having bond between them. It's called beta-1,4 glycosidic bond. So it does this by binding to the peptidoglycan molecule in the binding site first, and then reaction is catalyzed by the lysozyme, and this is the hydrolysis reaction. So hydrolysis is with the help of water actually. A water molecule is being added. So you need to remember this is with the help of one water molecule is being added to the single bond between the two adjacent sugar molecules. One is called NAM, and another sugar molecule is called NAG. So it is always in this order. So NAM will come first, egg will come later. Okay, so you need to remember the sequence also because you may be confused by NAM, NAG, NAG, NAM, or anything else. Like this. So suppose there are large number of, so this is a sequence of sugars A, B, C, D, E, F. Suppose this D and E are, this is NAM and this is NAG. So N-acetyl muramic acid and acetyl glucosamine. Okay, so it is going to cleave the this particular beta-1,4 glycosidic bond which is present between the NAM and the NAG residue. Now this NAM and NAG. So the two amino acids are very important in this catalysis. One is glutamate 35 and aspartate 52. Okay, so we know both are acidic amino acids, and they are critical in activity of this particular enzyme. So glutamate 35, first of all, so see here, this glutamate 35 from the lysozyme is acting like proton donor. So it will donate some, so this is the side chain of this glutamate. We know it is an acidic amino acid, so it will have COOH, it will donate some proton. So it will proton donor. So if it is donating proton, to this beta-1,4 glycosidic bond in the substrate, where the second aspartate or the second acidic amino acid, that aspartate 52, is going to stabilize this intermediate, which is going to form this carbonium ion intermediate. So suppose this is okay, so it will stabilize this. This is the second aspartate 52 actually, which is shown here. So this is aspartate 52. It will stabilize the oxonium ion or this particular carbonium ion intermediate until it reacts with a free water molecule. So also this is very important because this reaction is hydrolysis reaction and water molecule must be involved, extracting the hydroxyl group and freeing a proton that will bond to the glutamate 35 and leave the enzyme unchanged. So let's see into the mechanism. So it will donate, glutamate 35 will donate this proton. Okay, so if it's donating, it will act like a general acid. Then it will stabilize, aspartate 52 is going to stabilize this particular intermediate carbonium ion actually. So then a water molecule will enter somehow, and this water molecule, this is HOH, H will be donated to this glutamate 35, and OH will be added here, which will lead to the breakage of this particular covalent bond, so that enzyme and this will be degraded and enzyme will be freed. So this box is an enzyme actually. So it is having glutamate, it is having aspartate 52. So finally it will release this. So see here, this glutamate is regenerated. It is having the same COOH, and this is also regenerated, which is having a negative charge, and that's how this bond is, which is actually intact in the first diagram, is broken finally. So this is called beta-1,4 glycosidic bond. So this will be released out of this box so that this enzyme will remain unchanged and the lysozyme will be able to degrade your cell wall of the bacteria, actually gram-positive bacteria, to glycan cell. Fine, so that's all for today. We will discuss rest next class. So we have discussed the two major types of catalysis mechanisms. One is called journal acid-base mechanism, and the second is called covalent catalysis. So now we will start with metal ion catalysis, and we will also discuss some electrostatic catalysis, catalysis through the proximity and orientation effect, or the catalysis by the preferential transition state binding. So we are left with these some basic things for tomorrow's class. If you have some query, you may ask. Otherwise, we will meet in our next class. Thank you.